Okay, let's go ahead and get started, folks. The exams are not quite yet ready, so I apologize. I was hoping to have them ready by now. They should be ready by tomorrow. I will put out an email as soon as they're available, and you'll be able to pick them up in the BB office as you did the first one. So I will let you know as soon as I have that ready for you. I have posted the key to the exam outside my door, so you can take a look at that. The old key is also there if you need to take a look at that. It's underneath the one that I posted, or the new one. And I think I mentioned to you earlier that I did make a link on the schedule page to the older lectures, so if you want to be studying ahead for what I'll be talking about tomorrow, you can do that. So I am pretty much in good shape to finish everything up on time, and what I'm going to talk about today is the finishing up a few considerations with respect to glycolysis. This includes some regulation considerations and also entry of other sugars into the pathway. Then I'll talk about gluconeogenesis. So that's my two goals for the day. If I get through both of those, I'll be quite happy, because that leaves me 2 lectures or 2 hours tomorrow to talk about glycogen metabolism, and that's just about right. So, well, before the exam, we finished going through the reactions of the glycolysis pathway. And the glycolysis pathway is, as I described earlier, a central metabolic pathway, meaning it's central to most organisms. We find glycolysis in virtually every organism on the face of the earth. And interestingly, we see very, very, very little difference from one organism to another, both in the structure of the enzymes and also in the reactions catalyzed. So it obviously is clearly a very, very important pathway. One of the considerations we have in running the pathway is that the cells have to work always within certain constraints that are provided for them. And one of those constraints, as I have alluded to before, can be limiting oxygen. So if cells, for any reason, are limiting in an oxygen supply, they can be in rather deep doo-doo, as it were. And the reason for that is because the glycolysis system is totally dependent upon NAD. All right, so we see right here NAD is needed for that one oxidation reaction that we have in glycolysis. And cells absolutely have to have NAD to keep glycolysis going. Well, how do we get NAD? As Dr. Merrill will talk about next term, you make NAD by the process of electron transport. And electron transport requires oxygen. So if you don't have oxygen, you will not have a ready source of NAD. So when that happens, cells have to find alternate sources of NAD. They can't rely on electron transport to make it. And instead, what they do is what you see on the screen here. They actually take the NADH that's produced from the glyceraldehyde 3-phosphate dehydrogenase reaction, and they convert that NAD and NADH back into NAD by, in this case, making ethanol. This is what happens in bacteria and yeast. And the reason that they're making ethanol is because they've run out of oxygen. So this provides them with a way of remaking NAD. In our cells, of course, we don't make ethanol. It'd be a lot cheaper Friday nights if we did. But as a matter of fact, what we do is we make lactate instead. We convert pyruvate to lactate. And that reaction also converts NADH to NAD. So as a consequence of that, cells have NAD to keep glycolysis going. Well, why do they want to keep glycolysis going? Well, if they're a rapidly metabolizing muscle cell, then they want to keep glycolysis going because it is a very important source of ATP for them. So if you shut down glycolysis, then cells have virtually no way of making ATP, and they're in very deep doo-doo. So it's important then that this redox balancing that I'm talking about must occur in order to make NAD so that the overall pathway can occur. Now, what happens if cells don't have NAD? Well, what happens if they don't have NAD is that that reaction that requires NAD doesn't occur. And it doesn't take too much of an imbalance in order for that to happen, okay? Okay, so here I'll remind you now is the reaction that cells are requiring NAD. There's glyceraldehyde 3-phosphate, there's 1,3-bisphosphoglycerate, and there's the reaction that requires NAD to make NADH. We remember, of course, that the reactions have energetic considerations for them, and that the delta G we need to have for a reaction being negative in order for it to go forwards, and that one of the factors in the delta G equation is the natural log of the products over the reactants. What are the products? Well, the products are NADH and 1,3-bisphosphoglycerate. If those products start to accumulate, we are not going to favor the reaction going forwards. In fact, we're going to favor the reaction going backwards, and that would be a disaster for a cell that's needing energy, because going backwards is going to rob the cell of ATP, and it's going to require things for the cell to do. So other sources of energy for the cell, which the cell won't have. So that's why this redox balancing is important. It's called redox balancing because the cell is trying to make an appropriate amount of NAD so that it can keep this overall process going. Now, as I said, what you see on the screen is the reaction that occurs in yeast and bacteria. I'll show you another schematic about this in just a second. And that schematic is right here. So if we look at the things that can happen to pyruvate inside of cells, we see that pyruvate really can go to three different things. That depends upon the conditions. It also depends upon the exact type of cell that we're talking about. So, for example, the conditions and the cells that will favor the production of ethanol will include lack of oxygen and, bacteria/yeast cells. They will go and undergo that process on the left as a way of making NAD. We don't do that, although I'll tell you an interesting sidelight about that in a second. We don't do that reaction, but instead we convert pyruvate to lactate when there's low oxygen concentrations in the cell. And again, it's for the same reason. We're trying to make NAD. The third pathway for pyruvate is shown on the right, and it occurs in virtually all aerobic cells when oxygen is present, because when oxygen is present, there is abundant NAD. And in that case, pyruvate gets oxidized to acetyl-CoA, and then acetyl-CoA is further transferred to the citric acid cycle where it is used. So three different fates for pyruvate. Now, one of the things I always point out to students is if we look at the process of going from pyruvate to acetaldehyde to ethanol, I discovered that this last reaction of going from acetaldehyde to ethanol actually requires an enzyme called alcohol dehydrogenase. And we actually have that enzyme in our body. And so the reason that we can't make ethanol from pyruvate is we lack the enzyme that makes acetaldehyde. Okay? We don't make acetaldehyde. In fact, what we do is we take the ethanol that we are consuming, shall we say, and the reason we have alcohol dehydrogenase is so that alcohol dehydrogenase can run the reverse reaction and actually make acetaldehyde. They're trying to detoxify the effects of ethanol in the body. And it's actually acetaldehyde that gives you the hangover. So because you have the same enzyme as yeast do, but you're running the reaction backwards, you end up feeling bad after a night of drinking. Okay? And the yeast, of course, doesn't have that phenomenon, at least as best we can tell. Okay, does that make sense? Okay. All right. Let's see. Here's alcohol dehydrogenase. No, I don't think there's anything worth saying there. And when we look at ways that various microbes deal with lack of oxygen, some of them are obligate anaerobes. Some of them are fairly nasty organisms, and they go through and produce a variety of compounds as a way of making NAD to keep glycolysis going. So if you're in microbiology, you may have a bit of learning with respect to that. Here are some of the products that can be made during fermentation. And I should point out that the word fermentation simply means that the cell doesn't have oxygen going on. So our muscle cells undergo fermentation when they don't have oxygen going on. Fermentation doesn't mean just the production of ethanol. It simply means products that are made in response to low oxygen conditions. So different organisms produce different things. Of course, lactic acid is what we produce. Acetate can be produced in an unusual process from lactate by certain microorganisms, ethanol, a variety of things here, and I won't go through those with you. Okay, so the consideration then of the balance of redox state is very, very important because cells just have to make sure they've got sufficient quantities of NAD or they really are in trouble. Well, glycolysis is a central metabolic pathway because, as we can see, it provides for oxidation not only of glucose but also of other sugars which can be slightly modified in order to enter the glycolysis pathway. So you've already seen, for example, fructose is metabolized. It could readily be metabolized to fructose 6-phosphate. That's an intermediate in the pathway, and we see that occurring here. I'm going to show you in a second how galactose, in fact, can be metabolized in the pathway as well. And both galactose and fructose metabolism actually are very important from a human health perspective. At least the one that's clearly established is that of galactose, and I make an argument for you in just a bit that fructose is as well. Okay, well, let's look at galactose metabolism first. Galactose is, as I referred to earlier, milk sugar. And milk sugar is not is milk sugar, but it's a component of milk sugar. Milk sugar, of course, is lactose, which is a disaccharide comprised of galactose and glucose. So when you break down lactose that you get in your diet from drinking milk, eating ice cream, etc., you produce an abundant supply of galactose. Well, galactose, as I said, can be metabolized in the glycolysis pathway, and it takes a little bit of manipulation to have that happen. So there's a couple of reactions that happen. First, galactose is converted into galactose 1-phosphate, and the enzyme name there isn't really critical for our purposes. And galactose 1-phosphate can then go through this sort of cyclic exchange that I'm going to lead you through in order to be metabolized in glycolysis. It looks like a sleight of hand, all right? So you'll see what the cell is doing here in a second. The cell starts the process with two things. One is galactose 1-phosphate that we just made in the previous reaction, and the other is UDP glucose. I've briefly mentioned UDP glucose and told you that UDP glucose is an important substrate for making glycogen. It's also an important substrate for conversion of galactose. Well, how does galactose get converted? It gets converted in the following way. Notice this is a glucose and this is a galactose. All right? In the very first step, the glucose that was on here is released as glucose 1-phosphate. So notice the blue guy here becomes the blue guy down over here, and it is replaced by galactose. So whereas we had UDP glucose up here, we actually have UDP galactose down here. UDP galactose can then be epimerized. Now, I briefly mentioned the word epimer to you before. Epimers are sugars that differ in the configuration of only one carbon. And that describes glucose and galactose. The only carbon at which they differ in structure or in configuration is carbon number 4. It's this guy right here. So what this epimerase is doing is it's converting UDP galactose into UDP glucose. It's converting the galactose into glucose. All right? Well, we're done because we've now regenerated our starting material, which was UDP glucose, and we can accept another galactose 1-phosphate. So each time this cycle turns, one glucose 1-phosphate is released. Now, glucose 1-phosphate, as we will see tomorrow, can readily be converted into glucose 6-phosphate and metabolized in glycolysis. So by generating glucose 1-phosphate by this cyclic pathway, cells are able to metabolize glucose. Now, I usually have questions at this point, so I'll stop and take questions about what I've just told you. Does it make sense what's happening?
So you said the UDP galactose goes to glucose and to glucose and phosphate?
So UDP galactose goes to UDP glucose, right? And then UDP glucose comes back up here and starts the whole process again,
Okay.
right? So each time when we release a glucose 1-phosphate, it was the previous galactose that this guy had bound to, all right? The previous galactose it had bound to. There we go. Does that make sense? So I can say it looks like a sleight of hand that the cell is doing, but in fact, it's actually releasing glucose 1-phosphate each time around.
Wait, so is when the glucose is turned into pyruvate, is it using glucose 1-phosphate or the UDP glucose? Or both?
Well, I'm not sure what the pyruvate connection was.
I don't know.
Okay, so the main thing here that's happening is this is a cycle, and every time the cycle spins, it spits out a glucose 1-phosphate. It's taking in a galactose 1-phosphate. So the net effect is that it's taking in galactose 1-phosphate and it is releasing glucose 1-phosphate. If this acts up again, I will turn it off. Whoa, what happened there? Not good. Okay, there we are.
Repeat the part about the glucose 1-phosphate, how it can go back into, can go into glycolysis,
This can go ultimately to glycolysis because, as we will see tomorrow, there's an enzyme called phosphoglucomutase that will convert glucose 1-phosphate into glucose 6-phosphate. Then it can readily be oxidized in glycolysis. So as a consequence of all this, cells are able to use the energy of galactose just like they would glucose. Just like they would glucose. Okay, so take a look at that. I think if you sketch it out, you'll see it's not too much of an issue here. Now, from a human health perspective, I said there were human health considerations. There are two, actually. One, of course, is the phenomenon known as lactose intolerance. That doesn't directly relate to what I just showed you here. It relates to the problems that cells have or people have with digesting lactose. Lactose intolerance happens not because of that pathway up there, but rather because of the lack of, or the diminishing quantities of an enzyme known as lactase. The older you get, the less lactase you make. And that varies somewhat from one ethnic group to another. Asians tend to have lowered amounts of lactase compared to people from other areas of the world. And if you lack lactase, then what happens is the microbes get a hold of the lactose instead of you getting a hold of the lactose. And they produce prodigious quantities of gas and cause, in some cases, some very bad discomfort, let alone a significant amount of flatulence. So lactose intolerance can be a very painful disorder. The other consideration actually is right here. And the consideration I'm showing you on the screen happens when we lack those enzymes that do the conversion in that cyclic pathway that I just showed you. So if those enzymes, either of those enzymes is deficient, then what will happen is cells will accumulate galactose. Everything will back up to that point. So instead of running the glucose or galactose 1-phosphate reaction to make galactose 1-phosphate, galactose accumulates. Well, when galactose accumulates, the body tries to detoxify galactose because, like glucose, it's a poison. And the choice of weapon for detoxifying the effects of galactose is to use this enzyme aldose reductase, and it converts the aldehyde that we see here into an alcohol that we see over here, known as galactitol. The problem with galactitol is it can readily form crystals in the lens of an eye. And so accumulation of galactitol is linked to the formation of cataracts. And that happens again, partly as a result of deficiency of those enzymes. You can get cataracts by other mechanisms, of course, but this is one mechanism by which that can occur. Okay. Now, it's at this point I usually stop and do a little sidelight and talk about what I like to sort of jokingly describe as Kevin Ahern's theory of obesity. So I'll tell you. Yes, sir?
I was just wondering, if you ate less lactose products, could that prevent getting cataracts later on in life?
So if you eat less lactose, will that prevent cataracts? Well, it would if that were the only way of making cataracts. But there are other mechanisms that can generate cataracts. So if you were susceptible in that sense, that as you were lacking those enzymes, then I think one of the prescriptions a physician would give you would be, in fact, to do just that, reduce your consumption of lactose in your diet. But there are other ways of getting cataracts. Okay. Well, as I said, this is Kevin Ahern's theory of modern obesity. All right. Now, I don't know if you saw the newspaper this past weekend, but they had some rather shocking statistics about obesity in the United States. Colorado is the least obese state in the country, and the obesity rate in Colorado, as the lowest state in the union, is higher than any of the states had 15 years ago. So obesity is an epidemic, and a lot of people wonder why. So I'm hoping to give you one explanation for why I think that obesity is on the rise in the United States. Okay? It actually relates to glycolysis. And to understand this, we need to understand something about the regulation of glycolysis. Okay? So I'm thinking as I'm looking at this that perhaps I will come back to this after I've talked about regulation and show you why I think that regulation is the real problem that happens in the metabolism of fructose. And fructose is important consideration because the thing that's happened in this country in the last 20 years is the incorporation of large amounts of high fructose corn syrup in the American diet. And this, I think, is the root of the problem. But let me come back to that after I talk about regulation and tell you more. Okay. There's the lactose intolerance. There's the lack of the enzyme lactase. This is what lactase is doing. And you can see galactose and glucose arising from that. As I said, all of us as we get older tend to produce less lactase because over evolutionary time, we didn't drink milk when we got older. We drank milk when we were infants and then stopped doing that. So it's only in modern times, more modern times, that we have been consuming milk and milk products through the rest of our lifetimes. All right. Well, let's talk about regulation of glycolysis. Regulation of glycolysis is interesting and odd, I'll be honest with you. And it's odd partly because of the energetic considerations that we have for the pathway. All right. Here's an early odd observation. Okay. Now, I think I said them before, but if I didn't, I'll say them here. There are 3 enzymes in glycolysis that are important for regulating the pathway. They are hexokinase, which catalyzes the first reaction, and it's the one I won't say very much about at all. The second enzyme that's important for regulating the pathway catalyzes the third reaction. It's phosphofructokinase, and as I probably said before, that is the most important regulatory enzyme for the entire pathway. The third enzyme that is important for regulation of glycolysis is the last one in the pathway, and it's pyruvate kinase. And I'll say a little bit more about it later. Okay. The one I want to focus on right now, though, is phosphofructokinase, or PFK. Same thing. This, at first glance, may not seem odd, but in fact, it is odd. All right. Look at the kinetics of regulation of this pathway. All right. If I look at the concentration of substrate, fructose 6-phosphate, versus reaction velocity, so I'm basically plotting V versus S, I get 2 very different curves. One if I have low ATP and one if I have high ATP. Now you would look at this and you would probably say, well, that's fairly straightforward. We learned that when you have a sigmoidal plot, it means what with respect to the enzyme? Allosteric enzyme, okay? And in fact, it is being allosterically regulated by ATP. That's not odd. What's odd is ATP is a substrate of the enzyme. Okay? It's a substrate of the enzyme. Now, how is it that if I normally take a substrate for a reaction and I add large quantities of the substrate to a reaction, don't I normally increase the velocity instead of decreasing the velocity? That certainly happens with every other enzyme that we've ever talked about. How is it that when we increase the ATP concentration, we actually see a reduction in the velocity from here down to here? How does that happen? Well, the answer to how that happens is that the enzyme has two binding sites for ATP. One is a substrate binding site, and one is an allosteric binding site. Now, I'll let you tell me which one has higher affinity for ATP. The substrate binding site or the allosteric binding site? The substrate binding site has higher affinity for ATP because we never want to have a situation where we shut the enzyme down. Okay, we don't want to shut the enzyme down because this is an important reaction. In low quantities of ATP, that is in fact the time we want the enzyme to be the most active, because that's when we need to be going through glycolysis to be making energy so that we can make more ATP. Well, this works very well because at low concentrations of ATP, only the active site binds to ATP. The allosteric binding site is not, in fact, bound by the enzyme. However, when the ATP concentration starts getting high, the cell doesn't want to be burning up all of its sugar. It's already got plenty of ATP. So then the ATP binds to the allosteric site and starts shutting the enzyme down. So it's a very odd but interesting phenomenon. The enzyme is interesting in another way in that this enzyme is regulated by several allosteric effectors. Okay? Another one is AMP. AMP turns the enzyme off. I'm sorry, turns it turns the enzyme on. And that makes very good sense because AMP is an indicator of low energy in the cell. And low energy in the cell wants to be running glycolysis. It wants to be making or breaking things down so it can make more ATP. So AMP is an allosteric activator of the enzyme. Well, there's yet another allosteric effector of the enzyme that's even more interesting. And it's one that unfortunately students find kind of confusing. So let me talk about that. This allosteric effector is called F26BP. Okay? Now that sounds like an intermediate in glycolysis. It's not. All right? It's not an intermediate in glycolysis. F16BP is an intermediate in glycolysis. F26BP is produced, as we will see, by other enzymes. And F26BP strongly activates PFK. It strongly activates it. In fact, F26BP is present in such tiny quantities in the cell that it wasn't until about the 1980s that the molecule was discovered. Very tiny quantities of this were very strong, we're strongly activating PFK. Okay? Now that's represented in the things that you see on the screen here. You see relative velocity plotted against either one substrate concentration or the other substrate concentration. And what you see is that in both cases, when F26BP is present, as it is here or here, that gives the maximum velocity. And you can see it's at 1 micromolar. It doesn't take very much to activate it. Here's 0.1 micromolar. So very tiny quantities of F26BP are strongly activating phosphofructokinase. All right. Now I'll talk later about how that molecule actually gets made. In fact, I guess I'm going to talk about it in just a second here. So one of the questions I get is, well, how do cells make F26BP? It's actually made from fructose 6-phosphate, but that doesn't make fructose 2,6-bisphosphate a glycolysis intermediate. Okay? It's made from fructose 6-phosphate, but the only thing that happens to fructose 2,6-bisphosphate is it can get broken back down into fructose 6-phosphate, so it never enters the glycolysis cycle. But fructose 6-phosphate is used to make it. Right? So basically cells make it and they break it down. and the making it and breaking it down depend upon the conditions that the cells find themselves in. Just as a brief review, if you forget what fructose 1,6 bisphosphate looked like, that's what fructose 2,6 bisphosphate looks like. And it is, as I said, an interesting molecule present in very, very low concentrations in the cell. Okay. Now, when we look at the regulation of this enzyme, I'm sorry, not the enzyme. Oh, now, come on. Regulation by this enzyme. All right. What we see is, oh, they're both the same one. I was thinking I had that set up differently. Okay, well, let me come, actually, I know what will happen. I'll come back to it down here. It'll actually come up down here. So we'll see, the reason I've got it in gluconeogenesis is that fructose 2,6 bisphosphate affects both the enzyme of glycolysis, which is PFK, and the corresponding enzyme of gluconeogenesis, and it affects them in opposite ways. So let me come back and talk about that after I talk about gluconeogenesis. All right. Well, here is the last enzyme in the pathway. The last enzyme in the pathway is pyruvate kinase, and it is the third regulated enzyme in the pathway. As you can see here, this enzyme can be regulated in a couple of ways. One I already told you about the other day, and that was that fructose 1,6 bisphosphate allosterically activated. That was that example I gave you of feed-forward activation. That enzyme is also allosterically inhibited by ATP. Now, that makes very good sense if you think about it, because again, if cells have plenty of ATP, they don't want glycolysis going any further. They stop it in its tracks, and they stop it by stopping pyruvate kinase. In addition to the allosteric regulation of this enzyme, the enzyme is also regulated partly by phosphorylation, dephosphorylation. And the phosphorylation turns out to be controlled by our friend protein kinase A. One of the enzymes that protein kinase A can phosphorylate is pyruvate kinase, and that is how we make the guy on top. Making pyruvate kinase favors the inactivation of the enzyme. It favors the reduction in the production of pyruvate. And this is a very important reaction in muscle cells. The reason it's important is, if you recall what epinephrine did, it stimulated the body to start breaking down glycogen. I said we had that 7-TM, and we activated the G protein. We activated the production of cyclic AMP, which turned on protein kinase A. And I said protein kinase A would go and phosphorylate the enzymes of glycogen metabolism to produce glucose 1-phosphate. If it's producing glucose, it doesn't want to be burning it up. And the reason it doesn't want to be burning it up is because the liver is trying to make glucose for the body. So at the same time as it's breaking down glycogen, it's turning off glycogen breakdown because it doesn't want to waste the glucose it just made for the rest of the body. And I'll talk more about that tomorrow, actually. All right, so the combination of these two, that is the allosteric regulation and the covalent modification, allows cells to have very strong controls over the activity or the process of glycolysis occurring. The top pathway, which involves the phosphorylation, is a hormonal response. And the bottom pathway down here, these allosteric effectors, are related to the instantaneous cellular environment. Okay? So both of those play roles in controlling whether or not glycolysis is occurring. Okay, now I've talked about regulation. Let me come back and talk about our obesity epidemic. Okay, and as I said, this is my own pet theory. We'll see whether or not it's true. Oh, I keep going back to that figure. I must like that figure. All right, and where was I? Oh, it's up here. Fructose metabolism. All right. Now, when we look at fructose metabolism, I said that my theory is that we have an obesity epidemic because we started dumping high fructose corn syrup into our soft drinks. Why did we do that? That's the first question. All right. The reason that we did that had to do with import tariffs on sugar. So when sugar users had to pay increasing amounts for sugar around the world, they looked to local sources of sugar and said, wow, we are one of the biggest corn producers in the world. Why don't we use all of that starch that's contained in corn, break it down, and use it to sweeten drinks? And they did. Well, you think about starch, and when I told you about starch, what did we say about starch? It was a polymer of glucose, not a polymer of fructose. How do you get high fructose corn syrup out of that? Well, glucose, if you recall, is less chemically stable than fructose. So wanting their product to be stable for as long as possible, the producers used enzymatic means to convert a good deal of the glucose in starch of corn into fructose. So that's actually how you make high fructose corn syrup. Enzymatically, you convert glucose into fructose. It's not a complicated process to do. Fructose is chemically stabler and is fairly sweet tasting. So this gets us to the point where we see why we've got high fructose corn syrup that's out there in our soft drinks. So what does the effect of having that in our soft drinks do? My theory is this. The more fructose you eat, whether by eating high fructose corn syrup or just by eating a lot of sucrose, either way you can accomplish the same thing, your fructose levels in the body go up. Okay? As the fructose levels in the body go up, there is an alternate enzyme that your body has to metabolize fructose. And like glucose, fructose is a poison. So the body wants to do something with it to detoxify it, and in detoxifying it, it converts fructose into fructose 1-phosphate. So fructokinase catalyzes a reaction, as you'll see here. It's not unlike what hexokinase did, except for this is making fructose 1-phosphate instead of fructose 6-phosphate. Okay? Well, fructose 6-phosphate can be broken down by another enzyme in the body called fructose 1-phosphate aldolase. And if you recall the aldolase reaction in glycolysis, it splits F16BP into two pieces. Well, this guy splits fructose 1-phosphate into two pieces. It splits it into dihydroxyacetone phosphate. There's an intermediate in glycolysis. And it also splits it into glyceraldehyde. In the regular glycolysis pathway, we're making glyceraldehyde 3-phosphate. Okay? Well, we're lacking a phosphate here, so this is why we make glyceraldehyde. And you say, aha, glyceraldehyde must be the problem. And the answer is no, it's not, because glyceraldehyde can readily be converted into glyceraldehyde 3-phosphate. So what we've done is we've converted fructose into two glycolysis intermediates, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it seems as if everybody should live happily ever after. But wait, what we've done is we have bypassed phosphofructokinase. Phosphofructokinase is a regulatory enzyme. And that regulatory enzyme is capable of putting the brakes on things if our concentrations of ATP are too high. We saw that it was an allosteric effector of the enzyme. If we have too much ATP, PFK will be turned off. If we're not getting enough exercise, we have plenty of ATP. Now, if we have plenty of exercise and we have plenty of ATP and we are forcing the production of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, what happens to these two things? Well, in fact, they will force the next reaction, which is the production of a second glyceraldehyde 3-phosphate, which favors the production of 1,3-bisphosphoglycerate, which favors the production of 3-phosphoglycerate, which favors the production of 2-phosphoglycerate, which favors the production of PEP, which favors the production of pyruvate. We are pushing the production of pyruvate by eating excess fructose, whether by eating too much sucrose or by eating too much high fructose corn syrup, which people are increasingly doing. Well, why is too much pyruvate a problem? Okay? Well, the problem with too much pyruvate is you're sitting around, you're not exercising, you've got too much ATP. You have plenty of oxygen because you're not exercising. All right? Under these conditions, you convert pyruvate to acetyl-CoA, as you guys have already seen. Now you got a ton of acetyl-CoA. What do we do with excess acetyl-CoA? We make fatty acids. These are all established biochemical principles. We make fatty acids. Well, what do we make from fatty acids? You got it, we make fat. So, Kevin Ahern's wild and crazy theory about American obesity says that high fructose corn syrup is forcing the production of fatty acids and fat as a consequence of this reaction. Now, as I said, I have nothing besides my own theory to tell you that, so you don't have to go out and say, well, we've solved the fat problem, we know what to do. But I think it makes logical sense based on what we know about biochemistry occurring inside of our cells. Does this make sense? I'm Mr. Raving and Ranting today, I guess. Okay, so that is an important consideration in our metabolism. Okay, the last thing I'll talk about before we take a break are glucose transport proteins. I briefly mentioned these when I talked about insulin signaling. And if you recall, insulin signaling caused the movement of glucose transporting proteins called GLUTs to the surface of target cells. The GLUT, in fact, that I was referring to earlier with insulin is called GLUT4. And what a glucose transporter is a protein that is found in the cell membrane that brings glucose into the cell. It's an important consideration. Very, very important consideration because obviously cells have to get glucose inside of them when they need energy. And GLUTs, as you can see, our body has several different GLUTs. Some of them are specialized. Some of them are present almost all the time. Others are present in specific tissues for specialized purposes, as you could imagine. Well, glucose transporters are important for the next thing I want to talk about, which is an interesting phenomenon known as hypoxia. Hypoxia, as its name suggests, is referring to conditions of low oxygen. Now, these low oxygen conditions that I'm talking about are not the same low oxygen conditions that I was referring to earlier with respect to, let's say, exercise. If you go out and you go jogging or you go running fairly fast, for that period of time when you're running, your cells, your muscle cells, or at least the ones in your legs, may be relatively oxygen deficient. Hypoxia in the situation that I'm describing it here is more of a chronic problem. Okay? A chronic problem. And one of the places in which we see chronic hypoxia, okay, so I'm thinking about momentary hypoxia versus chronic, which is what I'm talking about here. Chronic hypoxia arises when we have, for example, a tumor that forms. Now, hypoxia can happen fairly normally. It can happen during growth and development. It can happen during exercise. But during a tumor development, there's something else that's going on. So tumors are, as you know, large masses of relatively undifferentiated cells. They are growing, in many cases, out of control. And what they are doing, because they don't have controls on them, is they are generally metabolizing things faster than non-cancerous cells are. Okay? Well, that means that they are using oxygen faster than the non-cancerous cells are. And their oxygen needs, therefore, are greater than those of non-cancerous cells. Okay? Well, one of the things that a tumor will do, if it becomes established as a tumor, is it will stimulate the growth of blood vessels to supply it with oxygen from the blood. Okay? But before that happens, what the tumor does, okay, is it's sitting there in a hypoxic state. Okay? It's sitting there because it's using oxygen way faster than the body can deliver it, and there's a long period of time over which this is happening. Okay? When this happens, the tumor cells, as your normal cells would do, starts making a factor called hypoxia induction factor, HIF, in this case HIF1. HIF1 is a transcription factor, and a transcription factor is a protein that will bind to the promoters of specific genes. So a transcription factor is a protein that will bind to the promoter of specific genes. So under these conditions where cells have low oxygen, whether they're a cancer cell or they're a normal cell, they will produce HIF, and HIF will bind to the promoter of specific genes and cause transcription of those genes to occur more readily. Now, what's really interesting is to look to see what genes get activated, that is, which ones does the cell start making more of. And that's shown right here. Okay? What happens? When the HIF factor is active, it turns on the following genes. Look at there, there's two gluts. Why is it turning on gluts? Because these cells have higher energy needs because they're not getting enough oxygen. So they start sucking up as much glucose as they can, and they do that by making more gluts in their membranes. That's interesting, but look at the enzymes below it. Hexokinase, phosphofructokinase, that's PFK, aldolase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, enolase, pyruvate kinase. This tells us that these cells really, really, really need to have glycolysis going. Why do they need to have glycolysis going? Well, I referred to the other day the fact that if we compare the ATP production in aerobic versus anaerobic conditions, that is high oxygen versus low oxygen, what we see is that in high oxygen conditions, each glucose gives you 38 ATPs. In low oxygen conditions, each glucose gives you 2 ATPs. To make the same amount of ATP in low oxygen, you've got to have 19 times as much glycolysis going on. You've got to have more glucose, and if you have more glucose, you have to have more enzymes to break it down. Now, this is an interesting phenomenon, and it's a phenomenon that a lot of people would like to target for killing tumors. Because if you can stop the production of these enzymes, or you can block the action of HIF in those target cells, you potentially have a way of knocking out tumors. And it may be a very effective way of knocking out tumors. And I actually know people who are working on just that phenomenon. Okay. It's time, I think, to take a well-deserved break. That's a lot of material. You guys are looking tired again. Let's do that, and I'll come back and talk about gluconeogenesis. Stop the camera briefly. Yes, sir.
Those